So the slides that we have up here, I think again there are 11 of them, will be the last bit of chapter 6. Next week we will kind of storm through chapter 7, and then the week after that, 2 weeks from today, is your next test over chapters 6 and 7. So it's just the 2 chapters this time. So, we're talking about the cytoskeleton to start out with. What does cytoskeleton mean? Wake up! What does it sound like to you? Cytoskeleton.
Structure of the cell.
Yeah, it's the cell skeleton. We're going to be talking about kind of internal structural components. Now, the next sections after this one, still in chapter 6, talk about some of your external structures. So we're going to get through all of this today. Now, there are 2 really common terms to know about the cytoskeleton. One of them is written up here, and it's this word right here, the microtubule. The microtubule. So whenever we talk about a cytoskeleton or the cytoskeleton of a cell, microtubules are a pretty important part. And I'm going to try to give you pretty simple definitions because we do have kind of a lot of terms today. These microtubules are simply hollow protein tubes. They're hollow protein tubes. That's what a microtubule is. And they are simply structural. They're like the steel beams of the cell. Okay? Because we talked about it. What's the outside of an animal cell? What is the outside of an animal cell? The cell membrane. This squishy, lipid-filled fluid structure is the outer barrier of your cells. Well, that squishy structure needs some support and some strength. And that's what the microtubules, or one of the things the microtubules do. Now, the other term, which unfortunately is not on this slide, but I'm just going to write it right here. There are microtubules, and then we also talk about microfilaments. Microfilaments. Those are 2 words that you will see in tonight's reading and the reading for Friday through the weekend into Tuesday. Microtubules, which are hollow protein tubes, and microfilaments, which are basically just these twisted protein chains. Twisted protein chains. So you essentially have tubes and chains, all made of protein, that provide for a general structural function. Microtubules, microfilaments. Here you can actually see a microtubule inside a cell that is taken from an electron microscope. So this is an extremely small structure that we would never see with the microscopes that we have here in school. Now, the video I show you later is going to show you a great animation of what I'm about to talk to you about. And this is related to these microtubules and the general function of the cytoskeleton. And that's this structure called a motor protein. It's another term that comes up a lot. And when you hear the term motor, what do you think of? Yeah, movement. That's right. You think of some kind of motion, movement, mechanical movement. And that's exactly what some proteins are capable of, and those proteins are known as motor proteins. These are tiny protein machines. They are machines. And what you see here is a motor protein right here. That's this that we're looking at in this section. And it almost has these two little feet, legs with feet. And they actually will walk along microtubules that are just stretched throughout the cell. And as they walk along, they'll carry a vesicle. What is a vesicle?
Membranous sac.
Yeah, just a little bubble, little membrane bubble. Membranous sac is a great technical term that your reading uses. It just holds something. So there are motor proteins that will carry and walk these vesicles from one place to another and drop off a package, drop off a delivery at some other location in the cell. Again, when you see the animation, it's going to look like a small, weird cartoon man carrying something, but this is a real protein machine that is right now acting throughout your body in your trillions of cells in thousands of different places in every cell. So it's not just structure that the cytoskeleton is used for. In a way, they can actually also act as kind of like a road system, like a roadway. Okay, so your reading will also talk to you about these two terms, centrosomes and centrioles. I want to talk to you about both of these right now and then move on. I'm not going to spend a whole lot of time here. A centrosome is just a region. And you can just see it's kind of highlighted in one part of the cell. Your book talks about how it's close to the nucleus, but you don't need to really know its location. It's a region that produces microtubules. So, microtubules are a very important part of the cell, very important component of the cytoskeleton. The cell has to make them and assemble them somewhere, and that's what's happening in the centrosome. You're kind of building the microtubules here. The centrioles are weird because we really don't know exactly what they do. Not every cell even has them. You have them in your cells. Animals seem to be pretty reliant on them because we can actually remove them and the cell will not function properly. But while centrosomes are the region where microtubules are made, centrioles seem to organize the microtubules in some way. If a cell doesn't have them, it'll make microtubules, but they won't be made correctly or at the right time or sent out to the right locations. And it's actually not clear what they do. I typically in a biology class wouldn't even talk about either of these structures, but in AP, sometimes they come up. So microtubules are important. These two structures simply direct their production. So let's talk about things that are more commonly discussed and honestly probably more pertinent towards an AP discussion. There are two other cell structures. These are a little different than a lot of the ones we've talked about because these are on the outside of the cell. They are the flagella and the cilia. Flagella and cilia. You definitely want to know what these are, the difference between them, and what they do. You're probably somewhat familiar with these terms. You may have heard flagella before. By the way, these are the plural, so the singular is flagellum and cilium, but that's not something that comes up a lot. So, for the flagella, these are long tail-like structures. They are long tail-like structures. And what do they help some cells do? Move. That's it. If a cell has a flagellum, it just kind of whips back and forth and allows for movement. This is not something you see in plant cells. You only see it in animal cells. And say in most animals, like you, the only cells that would have flagella would be the sperm cells in males, which they have an actual micrograph of that here. That's a very long structure for a cell to have. And it's simply for locomotion, for movement. Similarly, there are the cilia. These are shorter, hair-like structures. Typically also used for movement. There are definitely cells with cilia that use their cilia to swim around. But you have cilia throughout your respiratory tract, and they don't help your lungs move around, obviously. But what they do is they kind of create this brushing motion that allows you to get particles kind of filtered out and moved back up out of your respiratory tract. So they kind of help like clean your respiratory tract. Now, to connect these structures with what we were talking about at the beginning, your book goes into some painful detail about the flagellum and its internal structure and how you have these weird networks of proteins that form these different structures. There are so many terms up here that I'm not really worried about. Knowing the function of the flagellum is important. Being able to identify it as a long tail-like structure, that is important. But the reason I wanted to show you this is that the internal structure of these, some of these other structures are microtubules. So the flagellum itself, this tail, has these microtubules inside it. So this is a magnification of the flagellum, and you have these microtubules inside, almost acting like bones in the body. A structural... Why, Apple AirPlay? Why? So even though your reading will go into a lot of this, and it gets a little intimidating. They talk about basal bodies and these, again, kind of unique networks of proteins. All this, by the way, is driven by motor proteins that we talked about. So anytime you're talking about something moving, there's a motor protein involved. What's the energy source for these motor proteins? ATP. That's right. Yet again, that term comes up. And next unit we'll really get into that a lot more. But that's the flagellum, partially composed of microtubules. Okay, in your intestinal tract and in other places throughout other organisms, you have these extension, these hair-like cell extensions. They are not cilia. They are actually where the whole cell kind of like blobs out in this finger-like projection. You can see one here and 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 here that are called microvilli. If you put LLI, that's the plural, the microvilus word written up here is the singular. Again, in your intestinal cells, you have a lot of cells that have this kind of many finger-like extensions. They're not cilia. What would having all these finger-like extensions do for a cell? Think back to the lab we did last with the calculations.
Increase the surface area.
Yes. The whole purpose of this microvilli, of these microvilli, is to increase surface area. Just like the inner membrane of the mitochondria. We see that a lot in biology, where you have these finger-like kind of folded projections, either inside or, in this case, outside of the cell, that just gives the cell more surface. Why would your intestinal cells want more surface?
Digest quicker, faster.
It's not really about breaking the food down and digesting it, but doing what? After it's being digested.
Like getting it to leave.
It's not like the cilia of the respiratory tract where it's not involved in moving digested food along. Yeah, it's absorption. That's right. There are so many times where cells are absorbing things they need, and they do that by increasing the surface area. As digested food kind of moves and brushes along these extensions, you've got more membrane picking out what they want and bringing it into the cell. Now, again, to tie it back to some of the terms we've talked about, how is it that a cell can create this kind of protrusion? It's thanks to microfilaments on the inside that give it that structure. These long protein chains just kind of push the cell out into this shape.
Wait, so does the microvilli have the microfilaments inside of it?
Yes, there are microfilaments inside that actually push the cell membrane out into this finger-like shape. Again, cytoskeleton. That's a very skeletal function that you have here, is providing shape. So these microfilaments will provide shape for the microvilli, just like your skeleton provides your shape. I mean, you pull all your bones out, you're not going to have the form you have. Good question. Okay. Continuing on. Now, these microfilaments that give microvilli their shape, or microvilli their shape, you can say it either way, they are also used in muscle fibers. So you have these different filaments, and you'll see these terms actin and myosin. These are simply the names of the proteins that these strands are made out of. I would be familiar with them, but you don't need to know the difference between them. When your book talks about actin or talks about myosin, they are simply, it's the name of the protein that these chains are made up of. And these long protein filaments, microfilaments, they weave together in your muscle tissue, and they can actually kind of pull against each other, and that's how you're able to contract your muscles and relax them. It's kind of a neat, at the very top, I don't know if you'll be able to see it on the screen there, but muscles actually have what we call striation, these lines, and that is fiber after fiber after fiber, all woven in with one another, working kind of in cooperation to create these contractions. Again, one of the many functions of microfilaments and of the cytoskeleton in general. Okay, so moving on. This might not actually take as long as I thought. We're moving pretty fast. What we're looking at here is a diagram of plant cells. So we're plant cells have a lot of these cytoskeletal components. They have microtubules, they have microfilaments. They obviously don't use them to move like we use them for movement, but they are going to be used for structural components. But another structure, and again, this is kind of a separate section of your reading, so we're shifting gears just a little bit here, are the cell wall of cells. Now, as an animal, your cells don't have cell walls. Okay? This is not an animal structure. This is a plant and fungal structure, and a lot of bacteria, a lot of prokaryotes actually have a cell wall, but they're all different from each other in what they're made up of and to some extent what they do. We're focused on plants for this. So plants have a cell wall, and it is made of a polysaccharide that we have discussed before. Do you remember the name of that polysaccharide?
Cellulose?
Cellulose. That's right. The cell wall of plants is made of cellulose, which, if you're wondering how to spell it, it's literally cell, letter U, lose. Cellulose. Cellulose. Good. Now, your book goes into the primary and secondary cell walls and this lamella that you have kind of between different plant cells. I'm going to give you the very, very briefest of explanations here. The primary cell wall, when you see that term, that is basically young, flexible cell wall. Okay? Young, flexible cell wall. Still made of cellulose. Secondary cell walls tend to be what? Give me a guess here.
A little bit stronger.
Older and more hardened and stronger as a result. Yeah, I mean, they would definitely be more rigid. So the secondary cell wall tends to be an older, harder cell wall or structure. Now, just to relate that to kind of everyday life, lots of really young plants. You think about a tree, little tiny sapling. You see them out in the woods when you're walking around. You can bend them around. You can actually take trees and tie them into knots, and they'll grow with a knot like that. They are very flexible and pliable. But after decades of creating more and more cell wall, having those cells kind of crushed together because the plant's producing more and more of this cell wall material, and sometimes they even use chemicals to harden the cell wall. But after decades, a tree becomes much more structurally sound. And in fact, wood, the building material we get from trees, that is predominantly the secondary cell wall. So just because there's kind of an everyday association, I wanted to point that out. The lamella is a sugary layer between plant cells. So the lamella, which is this kind of really thin layer that they're showing here. By the way, each of these is a plant cell with the cell wall, and then the lamella is just this sugary layer that glues the cells together. Sugary layer that glues the cells together. Okay, and then to finish off this chapter, we have what's called the extracellular matrix. Extracellular matrix. I know that sounds like a really weird, complex term, but you got extracellular, which is one word, and then matrix. Now, there's a lot that's going on here. And I'm not going to go through every single term and tell you what everything does, but I'm going to give you kind of I'm going to try to get through these kind of quickly and give you just some terms to look at that you'll want to be familiar with. Just make sure when you do the reading over the next few nights or whenever that you get these down. Now, in general, what the extracellular matrix is, it's the space between cells. Okay? So you're trillions of cells all stuck together. But there is space that exists. I mean, it might be fractions of a micron, a micrometer wide, but they are spaces. Not all cells are absolutely jutted up against other cells. And that's kind of what the end of chapter 6 goes into. All the stuff between cells. Now, there is a term I want to write on the board first, and then I'm just going to point out some other terms. But a lot of these structures that we're going to talk about are made up of what we call glycoproteins. Glycoproteins. Now, obviously, you see the word protein in this term. So these are proteins. What is the glyco prefix reference? What other kind of biomolecule? glyco? What does that sound like to you?
Glycogen.
Like glycogen or glucose, it references sugar. Anytime you see that glyco, you're talking about sugar. These are proteins with sugars branching off of them that are bonded to the main protein. So it's kind of a sugar protein. It's a protein with sugars branching off. So it's a weird hybrid of two things we've talked about. And it gives them a lot of kind of unique properties depending on what they're trying to do. But glycoprotein, a protein that essentially has sugars branching off of it. Okay, so there's a lot of stuff that's going on in the extracellular matrix. So let's talk about kind of general purpose of the structures in this space. All these different fibers and strands that you see here, as chaotic as it looks, they have a few simple purposes. One, they can help provide structure to cells. Okay? That's a pretty standard function that a lot of these, like the cytoskeleton, the cell wall, all have. So all these filaments and fibers can provide structure. They also, though, help cells communicate. So it's not just structural. They also help cells communicate. And I'm going to give you say like 7 different terms that I just want you to kind of read about, point out, just to save time right now because I do want to show you the video. But some of these glycoproteins, as I mentioned, that serve these structural and communication functions are collagen. So maybe write down collagen. You can leave a space and just add in when you do your reading a brief definition for these. And we'll try to come back to them if we have time. You've probably heard of collagen, by the way. As you get older, your collagen breaks down, and your cells and tissues don't hold together as well, and what happens to you?
You get wrinkles.
Yeah, that's what wrinkling does. These are an enormous part of what holds you together. At least kind of skin to tissue underneath. So collagen, you see these fibers that give structure. You have what are called proteoglycans. These words. Here in green, you see these proteoglycans. They're just a glycoprotein, kind of a sugary protein combo. These strands just create basically a big tangled mass that allows your collagen to kind of set in place. Then we have this fibronectin, also a glycoprotein that anchors all these strands actually to the cell they are around. So we have structural support, kind of a web that holds that structural support in place, and then anchors that anchor it to the cell. Those are pretty simple construction techniques. Like if you wanted to build something outside of your cells, you would have structures like this. You probably just wouldn't give them these ridiculous names. So collagen, proteoglycans, fibronectin, and then finally integrins. Integrins, this word here. These are big proteins actually embedded in the membrane. And a lot of times they are part of the communication between cells. Now, for the sake of time, I kind of want to just leave it at that and then tell you about this very last thing. Again, this is in your reading. The last part of this chapter goes into what we call cell junctions. Basically, it's the extracellular space between cells, but it's kind of how cells are stuck together. And there are just 4 terms I'm going to have you write down. Read about this plasmodesmata. Write that down. They're basically just holes between cells that cells can kind of push fluids through so that some fluids can get from one cell to another kind of easily. Plasmodesmata. And then here, these 3 terms right here, I would just read over them and take notes on them in your reading. This tight junction, these desmosomes, and the gap junction. And I know I'm throwing a lot of terms at you, but I'm trying to just focus on a few of the many, many terms from your reading.
Can you repeat the?
Yes. On this slide, tight junction, desmosome, and the gap junction. And basically, with these tight junctions, desmosomes, and gap junctions, there's just one thing they can do. And it's essentially like seal the gap between cells, or anchor 2 cells together, or move fluid between cells. And it's laid out pretty clearly in your reading. But the tight junction, desmosome, and gap junction. You
What was the
have
last thing?
gap junction. Gap junction.
And then they seal the gap and anchor cells together, and then what else do they do?
Move fluids. The gap junction is going to move fluids between cells.
Oh, thank you.
So kind of seal, anchor, and move. I know that's a lot of stuff that we talked about today, but your reading, and this is 2 nights worth of reading that we just talked about, it's full of a lot of weird words, and I just kind of wanted to give you a little bit of focus on the vocabulary. There are a lot of other terms. If I didn't mention them today, and you can go back and watch the video, and you probably wrote most of these, hopefully you wrote most of these terms down. But if your reading goes into them, and I didn't talk about them on this part of the reading, it's not something we're going to focus on, and it's not, I don't want to say it's not important, but it's not important for our purposes. But that finishes chapter 6. So we're going to stop the video in just a second, but before we do, I'm going to show you a video that is on your coursework page. It's off of YouTube. It's linked on the coursework page, so you can watch it at home, maybe stay online, watch it while I talk about it. It's 8 minutes long. I just want to show it to you, show you how weird the cellular world is, and then explain kind of this small project that you've got a few weeks to get done. Explain the details to it, which are also explained on the task where the video is linked. So go ahead and stop the recording there for me, please, and we'll watch the